So today I'm going to show you some of the really interesting things. This is actually uh, the science behind a patent that we recently wrote called NOSE, NOSE, following our previous patent, SEE, sparse edge encoder. So at the end, I hope that I can really come explain that we have a grand project try to integrate three different jigsaw puzzles uh, concerning three different major inputs that how we actually interact with the external environment. So today we're going to talk about this. Okay, so some of the details of the pattern that I cannot release here, but then the science behind us that I would certainly be happy to engage all of you into active discussions. So if you just have any question, just interrupt me. Okay, so it's going to be um, casual, and I do hope that can stimulate some of the discussions from you. So the title of my talk today is The Dawn of Quantum Biology. So I'm Hsiu-Hau Lin from National Tsinghua University, and that's a little cute stuff I got for the open course wall. So let me see. Okay. So here's the outline of my talk. So I will briefly introduce what quantum biology is, and I will also give you one and two review article, which is sort of walk you through the why, because typically people think about biological systems are really, I mean, at room temperatures, there are lots of water fluctuations. Why would quantum dynamics ever evolve at all? And so, what exactly do we mean by quantum biology? And I do hope that the two examples that I showed today, the first one is photosynthesis. This is what we call that you actually observe coherent quantum dynamics. Whereas for the olfactory receptor, that is how we smell. If you think about this is actually something surprising. For instance, if you compare two molecules, H2O, let's look like a Mickey Mouse. And if you compare it to H2S, it's rather similar. And at room temperature, they actually rotate. So you don't even really detect this Mickey Mouse shape. How can we, typically most normal people that don't smell water, otherwise your brain would be too busy. And but then these two molecules actually make dramatically different olfactory responses. So why? And how did that happen? And so how do scientists understand this? So that would be the olfactory receptor. So here I'm going to provide you a rather interesting quantum aspect. I'm not saying it's right, but with our recent analysis combined with the AI ML, it seems that this might have a pretty good odds that it is the right mechanism that how we detect molecules. And so I will go through some of the theory. And so let me just warn you a little bit. The theory behind can be a little bit scary, but don't be too scared. Okay, so I'm willing to walk you through the major conclusions, and I would hope that I can skip most of the detailed derivation. And they are predictions, they're fun predictions. And then I will come to just the conclusion of my talk. Okay, so this is a very interesting review articles in Nature Physics. And so that's from Norris. So the title of the review article is very simple, it's quantum biology. Or one of the key themes in this review article is that can we find quantum coherence in biology? And so what exactly is quantum coherence? That would actually take us several hours to say, but I'm just going to really keep it short. How can one really understand quantum dynamics in one particular sentence? That is, the quantum dynamics only occurs in the phase of the wave function, which then gives rise a very interesting thing. That is that you might run into a very interesting situation that the wave function is changing, but the corresponding probability is not changing. And actually, that's why the quantum mechanics can explain that atoms, even though with dynamics, do not radiate any photons. Okay? So this sounds rather cool, but is it really possible that we can find it? So this review article sort of give you a very, very interesting example and also a primitive research. So the first one is photosynthesis, which I'm going to go through. And the second one is a very interesting thing. It's called magnetoreception. That is that if you think about that, how does a bird navigate and then keeping the orientations? And there are many microscopic mechanisms. One of them is that somehow, phenomenologically, we know that they have this magnetoreception. That is, it does not just detect the local magnetic field, it detects the derivative of the magnetic field. And so, how can you do that? And so, that's due to a very interesting radical pair with quantum entanglements. And then there's some other examples which you can also find in the review article. That's olfaction, vision, long-range electron transfer, and enzyme catalyst. Okay, so olfaction and the enzyme catalyst has some things in common. So here today, I'm mainly just going to talk about the olfaction. So let's then just get started. So the first one is photosynthesis. Okay. So why would we interested in photosynthesis? So let me just try to look into what happens here. When a light shines on a chromosome complex like this, the energy actually flows around and then eventually come to a layer, what we call the FMO complex, and eventually propagates. So the FMO actually form a network and eventually just go to the reaction center. What's surprising is that this route turns out to be extremely efficient. You can sort of think about the photon is like an energy packet into a room. And the room is full of FMOs with very complicated structures. And so it's just giving this energy package from one side to the other. If you propagate this in a classical way, and eventually you need to pass on this energy to the reaction center, the process would be slow. But somehow in experiments, it turns out that this is a really efficient way. So one plausible explanation for this is that this energy was not really given to one particular FMO. It is actually propagating quantum mechanicals in the FMO complex network. So in that sense, it's everywhere. So as long as this wave propagates with the slight detection of the location of reaction center, it just collapses. And then the energy then just transfer to a reaction center. And one can actually do simulation, and this is in fact very efficient. But come on, we're talking about real biological systems at room temperatures. So how can this be? We all know that for quantum system, it should be simple and it should be at low temperature. So let's just look into what this FMO complex is. So FMO, as I show here on the right-hand side, is a trimer. So it has three repeated units. And if you look at each monomer here, it contains seven pigments inside. So to some extent, you can sort of think about this. If I try to simplify it and model each pigment as just one location, so you then just have seven locations with electrons hopping around. And that will then be reduced to a 7 by 7 matrix. And this is a schematic way of how you really write these pigments. And I do apologize. This is not my notation. This is the notation in the Nature paper that for this slightly slanted label 1, 2, 3, 4, 5 that denotes the location of the pigment. For the bold solid phase, that denotes the energy levels. Okay. So first of all, what has been quantum here? This pigment contains some electron hopping around, and you can sort of imagine that on average, it's charge neutral, right? So when electron move, it will left with a hole. Then this hole would then carry relative positive charges, right? So electron has negative charge and this has positive charge. And because this is negative charge and positive charge, so the electron cannot really leave the holes that much. They would actually combine back and then form together. And for these complicated structures, we have a very cool name. We call it exciton. So that's how physicists excite. Basically, that for neutral things, you excite electrons. You left with the hole. The hole bind with electrons. So this whole excitation behave like a particle. So we call it exciton. So this exciton, and so because you have seven sides, seven pigment, so it can be described Hamiltonian by a Hamiltonian 7 by 7 matrix. You diagonalize the 7 by 7 matrix, you get seven energy levels. And even for a very simple monomer, this is already complicated, right? It has seven matrices. And you can actually say that what would be the most commonly seen or observed energy transfer? It turns out, for instance, if I follow this red line here, it would start here, oscillating between 1 and 2, which means that you would basically make transitions between level 7 and level 3. So you can see here, level 7 and jump to level 3, and then hopping to here, and then eventually hopping back to the lowest energy state. That's the 1 here, the segment here. So then you got here. And we would have another similar energy cascade flows like this green arrow, but then I wouldn't just go into the detail. If you're interested, I mean, this Nature paper actually, it's really a milestone in this study. It's worth reading. So how can we verify what I just told you? I mean, what I just told you seems like a quantum mechanics class. I mean, what I know that is correct. So let's see how we can actually measure this process here by a very, very simple things. So this monomer contains seven pigments. It's almost like a black box. And this energy level using the quantum mechanics, E equal to h bar omega, which means for specific energy corresponding to a specific frequency. So you can sort of imagine that suppose now you have a black box. The black box has some folk inside, the sound folk inside, with some specific frequency omega 1s. How are you going to, without opening up the black box, how can you detect this omega 1? So smart physicists come up with a way. Well, okay, physicists usually do things pretty violently. If you remember that how we really try to understand the atomic structure, the Rutherford basically just use high energy alpha particle to just make scattering, right? Here we're doing the same thing. I have a black box. I suspect there is a sound folk inside with specific omega 1. Being an impatient physicist, what would you do? You just skip the box. You give the black box a kick, right? And hoping that maybe I will excite some sound. So you just give it a kick and measure the output. And if you're lucky enough by measuring the output, then you can spot the omega 1. That's as simple as that. And that simple experiment has a very fancy name. It's called pump probe. So the kicking part is what we call the pump, that you excite the system. And then you use another laser light to probe the system. Okay. So what do we expect? Suppose now that in this little box here, in this black box, I have two sound folks, omega 1 and omega 2. And how can I detect that? Well, you can actually use resonance to do that. Why? You can send in an input omega, which I call omega tau, and then try to measure the output signal omega t. If this omega tau is not equal to omega 1 or omega 2, there's basically no output signal. So the signal is very, very small. But if this omega 1 tau happen to hit either omega 1 or hit omega 2, then it would generate this omega 1 and omega 2 signals in the omega t. So in this way, if I'm using the input, okay, the pump, omega tau, and the output, the probe, omega t, as the horizontal and vertical axis, I know that only these two spots around these two spots would have signals. And at all other things, there won't be any signal. So then it becomes relatively simple because I can change omega tau, I can measure omega t, and I just plot my signals here. And if you just find along the diagonal there are several peaks, then you know what's inside the black box. Okay, so that's really nice. So let's try to apply this to what we try to measure and see what happens. So in the papers, if you just look it up, they can actually do several different things. For instance, this 200 femtoseconds mean that you kick the box and then measure its response after 200 femtoseconds. And here, let me just concentrate on they are all similar and consistent. So let me just explain one here. So for this particular one here, this is when you wait for 1000 femtosecond. Okay, so that's pretty quick. Basically, you send in a strong laser light, excite all the excitation. That's almost like kicking on the sound fork. And then you send in a second laser light to look for resonance. And so you can actually see for the diagonal peak C and D, what we just expected, right, before. And in fact, if you then compare the value of C and value of D with the 7 by 7 matrix, you diagonally to get some number. And as a theorist or experimental, you'll be really happy here. You really find the set peaks of C and D, not only they appear in the diagonal, showing the resonant frequency, the location is also predicted correctly by the theory. But they are off diagonal peak. What happens there? Why would you measure something like this and something like that? How can we understand that? Okay, so that's the cute part. So that is the thing. See, what if inside this black box, these two sound folks, omega 1 and omega 2, are not independent? They are coupled together. And when two sound folks are coupled together, that means if you resonating with omega 1, you can also by this very weak coupling also generate an omega-2 responses. And so what they do then is that they can actually verify this phenomenon in this very beautiful, what we call the three pulse pump probes experiment. Basically, they send in three laser lights. For the first two laser lights, the first two laser light pump and entangles two exciton states together. And that's why basically you can think about that. You send it into pump. The first pump excite lots of exciton. The second pumps then entangle this exciton state together. So you get this entangle two exciton state, and you use the third one to probe what happens there. So these are the typical setup that we try to measure quantum entanglement. And so the realization of quantum entanglement is that then you will have this off-diagonal peak appear in the experiment. And that is indeed there. And once you know that, of course, you can also go to this much easier thing is you pump and probe, and the response will have this oscillatory behavior. And so you can see at lower temperature, the oscillatory behavior is better, while at higher temperature, this oscillatory behavior diminishes. And that also agrees with our common sense. So that's actually quite good. Well, except one important thing. You can complain that, okay, I got a very beautiful nature paper. I got another beautiful papers in science. And if you really follow this year by year, you would be amazed at how persistent these scientists are because they're starting from ridiculously low temperature and gradually push it up to 727K, 180K. That's still not room temperature. You might say that, well, now you've frozen everything at low temperatures, right? So you see all these beautiful quantum coherences. Why would that has anything to do with the real photosynthesis we observe at room temperature? So finally, after 2 decades of trial and error, that this in 2010, 2010, finally that we've observed this room temperatures quantum coherent oscillations with very complicated setup, but basically it has been verified that they are quantum coherent process in photosynthesis. I think in this way, this is actually something truly surprising that told you that if we really go back atom by atom to this very complicated FMO complex and zoom it into a monomer with seven pigments, it's still very complicated. And it's rather surprising that this very complicated biological structures can actually be captured by 7 by 7 matrix. Okay, 7 by 7 matrix might sound slightly complex in mathematics, but then in modern theoretical standards, 7 by 7 matrix is a piece of cake. It's very easy to handle. So that's the surprising part, and that has been verified experimentally. Okay, so that's the fun part of the photosynthesis. I think after 20 years of seek and search, that finally we found or we spot quantum coherence in something which is rather surprising. And let me just show you another interesting thing. That is the olfactory receptors. So the big question is the following. How do we smell? Okay. And first of all, we don't really absorb all the odorant molecules. Okay. So we actually have lots of odorant receptors. There are 27 proteins, and they are in fact in these epitheliums right above, inside your nose. And so Linda Bucks and Richard Axels discovered these olfactory receptor and eventually win the Nobel Prize in 1991. Oh, sorry. They discovered this in 1991 and then received it in 2004. So let's zoom in what happens there. So a little algebra, but of biology. Well, in fact, I'm a physicist. I shouldn't be explaining this. Maybe all of you know this very well. So this is a very simple physicist, naive way of thinking the olfaction pathways. So first of all, the odor molecule bind to the receptor like this. Okay. The second step is that miraculously, these olfactory receptor cells are activated somehow. Okay. And then we know that they're neuron cells. So then the current is passing through, and the signal somehow relays in glomerulus. Okay, so basically the same types of olfactory receptor, eventually the signal will wire up together in the glomerulus. And this glomerulus, the signal at glomerulus was processed and eventually sent to the high region of the brain. So you have several different things. And today I'm basically just trying to really concentrate on the very, very simple part. That is, how do we really read in the odorant molecule? When I say I smell coffee or tea or some really great apple pie, what exactly was my nose detecting? What is it? And I can share with you the mainstream's understanding of what has been detected. First of all, let's just look at the olfactory receptors. It is rather surprising that we have more than 350 functional odorant receptors. Okay, and that that's a little weird. In fact, if you really look at the statistics, that use up 3% of our whole genome. And in fact, that's second to our immune system. And for curious kids like me, I think it's a myth. It, I understand that why we spend a huge portions of genomes to decode different types of things for our immune system. Otherwise, you would die because we're surrounded by viruses and bacteria. And so we need that. But it's rather interesting that the second biggest genetic groups is actually the olfactory receptor, which is actually use up the 3%. And if you really treat it as programs, you then find it's coded in a very, very strange way because they all trans 7 proteins. So in terms of morphology, they're very, very similar and they're only little 20 subtle differences. It's almost like you're writing a program or try to describe the parabolic curve, how you threw a ball. And when you change the initial values, you should just change the initial value and retaining the whole program, right? But no, for each initial conditions, you write a program. And that is why it actually eats up this huge amount of information in our genomes. Okay? But we're not going to talk about that because I also don't know the answer. Maybe you guys know better. And so here is a very simplified version that our olfactory receptor is a G protein couple receptors. Somehow, when the odorant dock into the receptors, this alpha, beta, gamma get released, and then it generates this CM, and the CM eventually control this ion channels, and then make the olfactory receptors, make the olfactory neurons fire. And it's complicated, but even at the phenologic level, it is not easy because, for instance, if you just have three different odorants here and you have three different receptors, it's not like one to one. For instance, here the receptors respond to all three different molecules, and here this is only one, and here that's a response to two, but then with different strengths. So you can say that the smell it's actually a very complicated foreign language. And in fact, if you now have a very specific or very easy way to classify all odorant molecule, there would be actually something great. And so in order to do that, we need to first understand that how exactly is this odorant molecule trigger the neuron signal. Okay. And the current story or the mainstream story is what we call the shape theory or the lock and key model. It's basically saying that the odor molecule or the hormones or the neurotransmitter is like the key. A receptor is like the lock. And when the key and lock match, and then it clicks, and then there's some response. But then if you just keep asking, but then what happens there? Is that really because this molecule somehow collide with the receptor? That what exactly happened there? Then you get a little bit confused. Okay, so we'll talk about this a little bit more. And this, and so in order we can actually mimic this key. Basically, we can use like a pin with specific shape. We call it agonist, and you can in fact create a similar, not exactly the same not exact the same response, but similar responses. And they are also antagonists, which with similar shape, which means that this, for instance, this is like a branch, you can actually stick into this lock receptor, but it won't trigger any responses. And this is actually good. Think about that. If we have antagonist for smell, you can just spread that to your nose, and you can enter the bathroom and all sorts of things with unpleasant odor, and you wouldn't smell it at all. See, nowadays, because we don't really have very efficient antagonist for all smells, what we do is that we try to cover up the unpleasant smells by some other smell, typically lemons, and so that you won't feel that uncomfortable. And so one of the famous antagonists, of course, is coffees, and I think I probably don't need to think into too much detail. That is, if you compare the caffeine and adenosines, so our brain is actually basically why we get sleep is because that when you get tired and eventually you would emit the adenosine, which is an agonist. And this would actually trigger the signal, suppress all neural activity, and then you then have these sleepy feelings. And so suppose you have a report due with the tomorrow, so then you decide to just drink some coffee and then try to make the night. And so what exactly does that work? That does not really wake you up. You are still equally tired. But this caffeine has similar shapes to adenosine. So it acts as antagonist, which basically that this caffeine just combines with the receptors. So then these adenosines do not have the chance to trigger all these suppressions for the sleepy feelings and all sorts of things. And that's how that you actually hang there with all your tired body and responses. And so this is actually not very efficient. Okay, so that's actually how caffeine works and keep you sleepless at night. So then now go back to the olfactory receptors. So basically, if we do believe the major theories described, for instance, in one of these very nice and easy readings, review article in Nature, that just means that this odorant has specific shape. And this specific shape that work like complicated keys, which can then open up different sort of locks, the receptors. And that's how we explain the olfactory responses. But is that so? So let's just compare two interesting things. So this is fluorescence and this is nichrosens. So what is a fluorescent? Fluorescent is like a hamburg, okay? So the top layer is C5H5 and the bottom layer is also C5H5. So it's like the breast of the hamburger and it's stuck with ions inside. And so you combine them together, this is ferrocin. And another is called nichrosine. So you can sort of think about that. This is like Big Mac with beef inside, and somehow this is just another hamburger with pork inside. They should have exactly the same shape, right? And these are like nanoscale floating in air. And in fact, ferrocin and nichrosine, they smell differently. Normal people, you don't need to be an expert, okay? Yeah. You don't need to be an expert. They smell differently. Come on. So our nose somehow is so advanced, armed with this nanoscale molecular differentiation. If you really compute the difference of this ferrocin and nichrosine, as you can try to tell yourself, is that nichrosine are slightly thicker. And as a physicist, I don't really think we have this beautiful palm probes measurements in our nose. I just don't believe that. This is too amazing. Okay, maybe that's true. So this really opens up an interesting question is that maybe we do not just detect the shape. There's something else. And what would that be? So think about that. Suppose now you have a swipe card, which have passcodes inside. So it has a specific shape. When the shape is right, you can swipe it. But that doesn't mean that you can enter the building. You need to have the right passcode. So what is that passcode? Would that be the passcode is some sort of frequency that we talk about in photosynthesis with some energy level there? And so that's what we are going to look into. Okay. But before that, this whole idea of that there's some other messages, so quantum mechanical, somehow it's really due to this gentleman, Luca Turing's. That somehow the academies, or anyway, for people working on fashion, they just hate him so much for many reasons. There are really interesting story which I cannot go through with the limited time. You just look it up and you will find very interesting story behind it. So anyway, so the Andrei Teslas and Leslie Vassals, as far as I understand, is actually coming from the Linda Bugs group. And so they just hated these results. And so then they just wrote a paper in Nature Neuroscience. And this paper, this has really excellent abstract. See, at present, no satisfactory theory exists to explain how a given molecule result in perception of a particular smell. What does that mean? This means that nobody knows. See, this is a three-sentence abstract. I do wish that someday that you or me or we can all write these beautiful abstracts. The second sentence is that one theory is that olfactory sensory neuron detect intramolecular vibrations of the odor molecules. So what does this mean? This means that, so the first sentence is that nobody knows. And the second is that some guys claims he knows. And so the third one is that we use psychophysical method in human to test this vibration theory of olfaction and find no evidence to support it. So the third sentence just means that he does not know. And so that's the abstract basically. And in fact, the editor also write a brief introduction of this paper saying that if there are no such annoying so-called vibration theories, we wouldn't accept this paper to defend that these are really just nonsense. Okay. And if I look at the original paper of Luca Turing, yes, it does contain, because he's not a physicist, so the model he put up actually contained lots of mistakes there. But the whole idea is that maybe we are not just detecting the molecular shape. There's something quantum mechanical there. It's still there, right? Just because someone wrote a inappropriate model about that, that doesn't mean that idea is wrong. So let's just look deeper to see how are the other experiments is doing. So after Wasserhall's negative results, so there are a series of beautiful fruit flies experiments. So what do they do? So they basically take these normal airs and this ACP. So that's acetophenone, just something sweet. So the fruit fly likes it. So if you just put these two together, the fruit fly will be attracted here. But acetophenone has eight hydrogens around that. If now you try to just replace this hydrogen by deuterium, and so if you replace that, it is D3, and replace five of it, and replace eight of it, and you will find once you replace it with its isotope, the response is completely different. And in fact, you can compare this ACP and this DAACP that with equal strength. In fact, that you can see this disparity clearly. So what does that mean? That means that they are very strong isotope effect. But if all these shapes are really just bind to receptor by some chemical reactions, this doesn't make sense, right? Because isotopes, especially for acetophenone, which is not just hydrogen and deuterium that we are talking about, it's a bigger molecules. So replacing hydrogen with deuterium doesn't really change that. In fact, that fruit fly seems to hate this Da acetophenone that is fully deuteriated acetophenone. So why is that? And what's even more is that you might think, that, well, that's just because you have a bunch of dumb fruit flies. So there are some other experiments taking out the glomeruli of the bumblebees and then just repeat the experiments. So there are lots of details. I'm just trying to show you that for four different molecules, for instance, this is acetophenone again, the H just means that it's all hydrogen, and the Da means that it's just replaced with Da. You start to see differences. And what's even more interesting than in OCT is that here, you sometimes even see for this glomerulus that the response here is the largest, while here, this is actually negative. So it's a complete change sign, and you cannot just compare. So I think whether you like it or not, there are experimental evidence is showing that the isotope effect exists. And we probably need to explain that why it is so. So we do have a rather complicated theory, but today I would just briefly walk you through with a very interesting model proposed by Stoneham group that was published in Physical Review Letter back in 2007. So the idea is the following. Suppose now you have these really, really complicated things. So what we are going to do is that we are going to model these odorants later on by quantum chemistry because they are smaller. So we're just going to use quantum mechanics to solve them completely. But for the receptor, it's hopeless. So we are just going to get smart and then build a model. So the model we try to build, first of all, is a receptor. So it's an on-off switch. So we need to have a switch. So then you have an on state and off state and a turn between them. So in real, without any other dockings, the reason why they cannot turn on is really because the off state energy and the on state energy are not the same. So if you tunnel from off state to on state, that means you excite this receptor, then the energy is not conserved. But if now you have an odorant molecules, and the odorant molecule can oscillate actually, and this oscillation energy, just like in photosynthesis, we said when the charge move around and it left with hot and they bond together, and that quantum object has a name called excitons. Okay, physics like ONs. That's basically the particle of excitation. And so that's exciton. And so what's the molecule? For a molecule, you have a ground state configuration. If you just squeeze it a little bit, it takes some energy. And this energy is not continuous. It's one quanta, two quanta, and three quanta. And so that particular energy quanta and corresponding just like a particle, and we call them phonon. And in fact, the classical limit of phonon is nothing but sound wave that now we are using to communicate. Okay? So phonon is just a more fancy way of saying sound waves, basically. And so and all molecule oscillates. If you just stretch it a little bit, it would just oscillate. And in fact, luckily, we can use the Gaussian, the quantum chemistry program to calculate all sort of vibrational spectrums of these molecules. And so the magic happens. If then this oscillations is quantized, right? So it has specific energy. If this energy match with this, then this whole quantum process can occur. That means that starting from the off states, it then hop on to the on states, and the energy differences then release to this molecule, and this molecule just vibrate away and eventually dissipate these energies to some other collisions. And so then everything go back to normal. And so this actually makes perfect sense. So after they build a model, and they have actually really funny things, I encourage you to read it, at least find these funny comments. That's actually how I get interested in these projects. I was constantly at that time really reading quantum biologies, and I got annoyed by his comment. He built the models, and then how did he solve it? It just said that after standard approximation, with no references at all, the turning rate is this. I said, oh, okay. I got a PhD. I'm a physicist. Standard approximation. That should be quite easy, right? So what happens is that I chew on the literature, and I couldn't really derive what he means. That how do you really derive that? And so I got upset. So I stepped out, write the program, participate in a very interesting smell workshop at KITP, the very beautiful town Santa Barbara. And finally, after lots of calculations, we got a different formula. And this formula is actually rather scary. It's using the Fermi golden rule with Poisson and also the green function technique. So then we can actually lucky enough to get the exact solution. And in fact, even up to this day, I still do not understand what Stoneham's standard approximations because I finally got the exact formulas. And if I then take semi-classical level, yes, I did get that formulas after standard approximation. I do not think this is standard approximation in any major anyway. So finally, we got a formula, which means give me an odorant, give me a receptor. I can actually calculate how it responds. And it's cute. So that's finally we got this acetophenone. So what we do is that you can now see that this is the structure, the molecular structure of acetophenone. So you can count 1, 2, 3, 4, 5, 6, 7. I'm sorry, the eighth is hidden behind. So you got 8 hydrogens. So what we do is that we calculate the response of the acetophenone, and then it's deuterium versions. And so what happens? Well, then you basically got a signal of the blue lines and the red line. So they are actually different, which means if the receptors detect this particular passcode here, then you will get signal here, but no signal here. Or if it had passcode here, then you get signal here, but then no signal here. So that's actually really cool and really interesting. And since the deviation occurs at this particular range, I can actually go back and check that what modes is talking about. So this is the really cute part. So basically that the difference of this home and this home is this. And now you know that why the isotope I mean, happens there. It's this out of phase oscillation of two hydrogen atoms. And so it's not so easy or it's not so simple harmonic oscillator. It's basically just twice of the simple harmonic oscillator that you learn in quantum mechanics. And if you replace this hydrogen atom with deuterium, the mass just times two. So it changed the vibrational frequency. And because E equal to h bar omega, so it changed the energy. And so everything changes. And that's actually the origin of it. So in fact, not only you can spot that there are isotope effect, you can actually see that which vibrational mode, which distortions is really making the differences. And so it's this universal. So we try out another odorant molecule, it's called acetaldehyde. And we also see that the differences is there. And so what is that? So that's like a little doggies, and then this is like the tail oscillation modes there. So that's pretty cute actually. So we tried basically more than 50 odorant molecules, and they all showed this. So basically, if if you have particular oscillation modes which contain this hydrogen atom stretchings or oscillations or rotations or whatsoever, then it's deuterium version which shows significant differences. But then for other parts, then it does not. So whether it has isotope effect or not, that really depends on whether your receptor detecting signal at this frequency domain or in this frequency domain or in this frequency domain. And because of this, we now know that if we really go back to the previous one, that people often just think that, oh, molecule has specific shape, and then the receptor has also specific shape and depends on, for instance, this is like a square and a triangle, and thus it would then just have stronger responses for this receptor and for this receptor, and so on and so forth. And maybe that's not the whole story, because this story doesn't explain the fluorescence and necroscence that I just showed you, right? It's like two hamburgers with almost exactly the same shape, except the meat, one is beef, another is pork. And so if you then just say, what about we just apply our theory and calculate the spectrum differences of the nicholson and the fluorescent? Are they different? Yes, they are different. Why? Very simple. Because we are talking about how you squeeze the hamburger, right? So the iron and the nickels atoms has different mass. So they will corresponding to different frequency, and so they will show up differently. So basically, now we are proposing another way of looking at the odorant detection. That is, the shade must be right. For instance, you are swiping your car, right? You need to basically have a car. Using your pencil doesn't really break into the swipe card systems because it doesn't work. So you do need to have the shape. But just got the shape right doesn't really trigger these whole things. And with this model, not only it sort of explains some of the puzzles, it also provides the dynamics. See, with the shape theory, sometimes if you really ask yourself, going beyond the cartoons, what exactly trigger when you said that the odorant duck with the receptor, that everything happens. So what happens there? Why is this duckings so magical? But if now we know that this docking induces quantum tunneling, then it makes sense. Because quantum tunnelings then change the receptor from off state to on state, and thus the morphology change then created the cascades of the alpha, beta, gamma proteins, releases, and all sort of things. And so far, so let me just conclude. Yeah, sorry that I go a little bit over time for 2 minutes. So I think it's quite surprising that quantum coherence is actually spotted in biology. And the second thing is that I think the serial olfaction is still not yet complete. And this odorant detection, I somehow feel it has great potential because if you look into that, whether the similar mechanism can be spotted in some other biological system. And yes, in fact, that the G protein coupled receptors explain most of how the medicine works within our bodies. So this is going to be something important. And if we can, and for the poor guys, Luca Turing, who was originally the appointed in University College London, he eventually got fired from the college. And he also write a very interesting short article basically saying that the academy will take 10 years or 20 years to catch up to prove that his idea is right. But in any case, later on he used his theory to produce fragrance and make a big fortunes in Florida. So after all, still quite happy. Well, last time I met him, he was happy. And so what we do is actually really simple. Even though with all this very complicated thing, what we do is that we take the mainstream theory, the shape theory. We just ask, what if this is not a classical theory? It's a quantum theory. So what we mean is that we just quantize the shape theory. So it's almost like taking a spring and a mass and treating it as a quantum mechanical system. Then you get interesting results, which is drastically different from the classical result. And those are the results that I just showed you. And surprisingly enough, by quantize the shape theories, you got predictions similar to those obtained from the vibrational theory. So in our point of view, the shape theory and vibration theories are not really uh, competing with each other. They're actually the same, or in technical term, they can be described by the same metrics. It's just whether you go into the classical limit or you go to the quantum limit. And finally, this isotope effect, this olfaction profile, and there's lots of interesting things that one can explore in the future. Yeah. So I guess that I would just conclude my talk here. So thanks.
Thank you. It's very interesting talk. So we still have time for question because the class goes from 9:40 to 10:40.
Oh, okay,
Yeah,
great. Excellent.
you were doing very well on the time. So, questions from the audience?
We'll be happy to take any questions from you. Okay. And yeah, if not, let me just say one little thing to end this. So this exact formula turns out after taking some limits can really go back to Stonehams' original formula like this one. And if you look at this one and compare the Fermi's golden rule you learned, you found they're actually similar. Think about this. If you take 4 pi k T lambda as a parameter, eventually if you take this to be really, really small, this whole expression then turn into a delta function. And this then turn into energy conservation. And so to some extent, basically, the Fermi's golden rule still works. But then the receptor sit on epithelium. So it sort of broaden this delta function into this exponential form. Because if you plot out this exponential, it's a softened version of the Dirac delta function. Okay, so what does that mean? We were lucky enough that now you can sort of think about that. I just construct, just like Netflix, right? Netflix, they have this huge database. So on the road, this is like audience A, audience B, audience C, and audience D. And on that, then you have like different movies. And then there will be like stars or positive 5 to negative 5 and things like that, right? So you have matrices. You can think about that. We can also use this formula to build similar thing. You have like 350 receptors. You have about 1,000 odorants. So you have a huge matrix, and you just plot it out with all sort of things. And we ask ourselves, is there an easier way that you can understand this or not? And this is certainly beyond the what we call the conventional modeling, because not even multi-color simulation can help more. And so what we found is that there is a hidden structure that all the quantity here are non-negative. And there is a algorithm called NNF. It's called non-negative metric factorization. And because I construct a matrix, its elements, the responses are always positive. So I can actually use this algorithm to decode the whether we have very specific pattern. And with that pattern, then I can classify my molecules into groups. For instance, all this group is called fruity group, and these are called the ocean group and things like that. And this sounds like a really crazy, wild idea, but then we just finished it this year. So far, the successful rate is 100%. So
That's cool.
something is there.
We have a question for Laura in the chat.
Yes.
So, does it make things different if it's hot versus cold? And how is there an evolutionary reason that why these can benefit, like why the vibration theory can benefit animals and any animal
Excellent.
needs?
Excellent. Yes. See, I think this is actually excellent questions. And so I'm not trying to attack the conventional theories and saying that, oh, you must learn quantum mechanics. And but think about this. When you smell a coffee, it doesn't matter whether which angle you tilt. But if you really stick with the shape theories, the flow actually will affect the response rate, which means that if you smell it with different angle and all sort of things, you should detect the differences. But that that does not really our common experience, right? So there are some robustness of how we detect the smell. That once you duck in, something just happens. So you can ask yourself, what is the most robust mechanism we should lock this in? Well, it turns out it's very simple. If you have a hydrogen atoms, it's ground state energy. It's a specific number. It doesn't matter that how you actually measure it. And so that's basically the same thing here. So if there are some evolutionary advantages, that just means that if somehow this mechanism sits and start to work, it would just basically remain because it can actually wash out lots of minor genetic fluctuations and also the detection environment and all sort of things. It does has this, so basically that's the quantum robustness which will protect you. So let's go back to quantum mechanics. I believe probably Professor Lee has told you that for a simple Harmonic oscillator, if it is classically in the equilibrium point, even an infinitesimal force will then just make it jiggles, right? And depends on how you move it, it just moves differently. But in quantum mechanics, it sits in the ground state. It takes a minimum energy h bar times omega zero to get it excited to the first excited state. So that's what we call the quantum robustness. You just need that specific energy, otherwise it won't respond. And that's similarly, there's a similar mechanism here. Yeah, so that's great. I, it
Thank you.
seems that I also got some others. Yeah, and also that we smell differently in hot and cold. And the answer is that because the temperature used here is absolute temperature, this is not the Celsius or the Fahrenheit. And so then the temperature gradients that we can tolerate doesn't really make a difference. But what I didn't show you here is that the media actually has a very strong effect. So you will smell differently in water, in different solution, and also in air. And that also happens there. So that's another really interesting question.